And grand rounds. It's really my pleasure today to introduce Dudley Lamming. Dudley is a graduate for his undergraduate degree from MIT in chemistry. He then took a PhD at Harvard and then undertook postgraduate work in the lab of David Sabatini at the Whitehead Institute. During that time, he served as an instructor at Harvard and then took his first full faculty appointment just in 2014 when Vince Crines recruited Dr. Lamming to become an assistant professor. He also serves as a research scientist at this VA hospital and is co-director of the mouse metabolic phenotyping platform for the University of Wisconsin. He's also a member in affiliate programs, including the Interdepartmental Graduate Program in Nutritional Sciences Training, as well as the Cellular and Molecular Pathology Graduate Program, the Comparative Biomedical Sciences Graduate Program, and the Molecular and Cellular Pharmacology Graduate Program. He's published widely in high-impact journals, now with 30 publications, 4 book chapters, and 1 patent. He's been well supported. He arrived actually with a novel K99R00 grant from the NIH, which he's in the R00 phase. Currently, he is also receiving support from the American Federation for Aging Research, the Progeria Research Foundation, and he has a Glenn Award. In terms of teaching, he's active in terms of classroom teaching for endocrine as well as nutrition, and he's even lectured internationally in 2015 in Copenhagen. He's been active as well in mentoring and education, listing mentoring for a Career Development Award here at the VA, postdoctoral and graduate students. He's been involved in training for 6 undergraduates, and he sits on 4 thesis and postdoctoral mentoring committees. He's a very good citizen overall. He is serving as Director of Research Education for Endocrine. He's serving on the admissions committee for the Cell and Molecular Biology Graduate Training Program and is a member of the Research Education Committee here at the VA. He serves on 3 editorial boards and is associate editor for Nutrition and Healthy Aging and is a grant reviewer for the Biotechnology and Biological Sciences Research Council in the UK, as well as the Nathan Schock program. Along the way, he's earned a number of awards, including a Whitehead Institute for Biomedical Research Appreciation Award and an Anatomical Society Aging Cell Best Paper Award in 2013. And he's given a number of lectures. And this is a common theme you see here, where our own faculty are among the most impressive of the speakers we have each year at Grand Rounds. He's recently spoken at Cold Spring Harbor, Keystone, as well as regularly speaking at the American Aging Association and Gerontological Society of America, as well as speaking internationally. So we're very fortunate, first of all, to have Dudley Lamming as a member of our faculty, and please join me as in welcoming him as he presents Grand Rounds entitled Beyond the Calorie and Towards a Sweeter Life. Welcome, Dr. Lamming.
What should we eat? If you are a laboratory mouse, it's very easy. You eat the food that has been provided to you by your caretakers and research team. But as humans, we have a lot more options. We can eat, just this morning, for instance, muffins, bagels, or donuts. And noted food author Michael Pollan refers to the wide range of food options that humans have as the omnivore's dilemma. And when we think about all these food options that we have as opposed to laboratory animals, it's very perplexing because we now have an option to eat both healthy and unhealthy foods. And so Michael Pollan put it that we should really be eating food, not too much and mostly plants. And certainly I don't think that anyone could disagree with that type of nutrition advice. Unfortunately, that's not the situation that we have in the country today. Instead, we have a situation where, due to the abundance of high-calorie, nutrient-dense food that happens to be extremely delicious, over the last 20 years, there's been a dramatic increase in the incidence of obesity in the country. It's been called an obesity epidemic. And in its wake, there's been a vast increase in the percentage of Americans with diabetes, particularly type 2 diabetes. And this is concentrated and particularly severe in the southern half of the country, but Wisconsin has definitely not been unscathed. In Wisconsin, roughly a quarter of the population is obese, and almost 500,000 people in Wisconsin have diabetes, the vast majority of that type 2 diabetes. And 3 times that number, almost 1.5 million, are in a pre-diabetic state. And this is particularly of concern due to the fact that we have a graying population both nationally and in Wisconsin. And as we age, we tend to get much more diabetes. So a quarter of those over the age of 65 currently have diabetes. And as this pre-diabetic population ages, this is going to be a serious burden on the healthcare system and on the individuals concerned. This is also very severe in specific groups, such as American Indians, 40% of whom in the state of Wisconsin have diabetes. 20% of African Americans and other minority groups are also diabetic. Since we're in the VA hospital, it would be amiss of me not to mention that, again, just like the elderly, about a quarter of veterans have diabetes from a variety of causes. This includes both overnutrition as well as actually damage to the pancreas from their service. So instead of focusing on caloric intake, we all know that for the most part we should be eating less. Today I'm going to focus on moving past just caloric intake and focusing on the composition of the food that we eat and how that may affect our metabolism. So I'm going to tell us first a little bit about the big question, how much protein should we be eating? Then I'm going to take you through some data in which we look at human and mouse studies of reduced protein diets. I'm going to talk to you about the insulin sensitizing hormone FGF21 and its possible involvement in some of the phenotypes we see. And then I'm going to tell you basically what our current state of knowledge is and how we're going to continue to advance that. So it was the French that said, we are what we eat. And what we eat, since we have all these food choices, is heavily influenced by dietary advice from the government. For over 100 years, the USDA has been putting out various food guides. This is the last of the food pyramids. It was published in 2011. It was decided that that was too confusing, and now we've moved on to the My Plate, but it's much less visually interesting. And food can be divided into all of these various subcategories, for instance, fruits, milk, meat, and beans. But really, food can be thought of as being in 3 major categories in terms of its macronutrient composition, and those are protein, fat, and carbohydrates. And to simplify things a little bit, for many years now, both the USDA and various other nutrition groups have suggested that we should be reducing the percentage of our diet that comes from fats and carbohydrates. Primarily in the case of fat, this is due to the fear that fat is associated with cardiovascular mortality. And from a carbohydrate standpoint, the fact is that sugars and refined sugars give us a lot of calories without being satiating, so we don't feel full. However, if we reduce the portion of our diets that come from fat and come from carbohydrates, that implies that we should be expanding the percentage of our diet that comes from protein. And indeed, that is the case right now. So it's believed that Americans average about 16 to 17% protein in the diet. Whenever I refer to a percentage, that's the percentage of calories from the source. And it's recommended that we should be increasing that to probably up to about 21%. The Institute of Medicine suggests that anywhere between 10 and 35% protein is totally acceptable for the average adult in the United States. So how much protein should we really be eating? And a lot of data has been come about over the last 10 years in particular that suggests that this recommended protein intake is actually probably bad for you. And so one of the studies that came out most recently, this is a study that came out in Cell Metabolism last year, showed that lower protein diets were actually associated with decreased risks of cancer and decreased mortality in those under the age of 65, suggesting that we should really be reducing protein intake for a variety of reasons. A study a few years prior to that stratified individuals by protein intake, and the difference in diabetes risk between the lowest and highest protein intake was over twofold. And so protein intake was clearly associated with type 2 diabetes. And finally, a study a number of years ago looking at what happens to people who try and follow really high protein diets, as is suggested for various fad diets, found that higher protein intake was associated with increased mortality. So this all suggests that high protein intake is probably bad for you, and that lower protein intake may be good for you. But it's actually very complex, because when we as humans choose lower protein foods or higher protein foods, we're making a lot of other choices. And so, for instance, the USDA suggests that we could substitute lean meat for fatty meat and white meat for red meat. And so these types of choices really confound human studies. So taking a step back and going into mouse models, a group at the University of Sydney published a great study last year in which they fed mice one of 25 different diets. So this is a very large-scale study looking at low, high, and medium protein, carbohydrate, and fat intake. And so basically using the same sources of those diets and then mixing them up in different percentages. And what they found was that the mice that lived the longest were the ones that had the lowest protein intake. And what's really surprising is this is true even if almost all of their remaining calories come from sugar or all of their remaining calories came from fat. So we wanted to know if these types of interventions would have beneficial metabolic effects on humans. And to do this, we collaborated with Luigi Fontana's team at Washington University in St. Louis. He was looking at protein restriction in the context of prostate cancer. And so he was conducting a study in which humans were randomized into either a control diet group where they continued to eat their normal diet but kept food diaries about what they were eating, or a protein-restricted group in which the protein content of the diet was lowered for 1 month, such that they ate between 7 and 9% of their calories from protein. And so this group was provided diets by the clinical study. And so one of the things that is important to note is this is a very moderate degree of protein restriction. So the recommended protein intake for humans of this age and roughly of a roughly normal weight was about 56 grams a day. And so the protein restricted group was actually getting more than the minimum daily recommended intake of protein. So they're getting less protein, but it's still in a biologically relevant, translatable, and sustainable level of protein. So what did we find looking at physical and metabolic characteristics in these individuals? So this was a 1-month study, and we saw that there was a significant decrease in body mass index in the group in just 1 month that was fed a protein-restricted diet. And this was driven by over 2.5 kilograms of weight loss. So almost 6 pounds of weight loss. And slightly more than half of that was due to reduced fat mass. And so using imaging, this actually seems to be really driven by a decrease in visceral fat, which metabolically is sort of a worst type of fat that you can have. So a protein-restricted diet seems to have really good effects on body composition, and this is in just a very short period of time. So we're really interested in looking at glucose metabolism. Oh, and yes, I should mention that although the original intention was to have this be isocaloric so that everyone was eating the same amount of calories per day, it turns out that the protein-restricted group was actually eating about 5% more calories. So they were eating more, consuming more calories, yet they lost weight. So we were really interested in glucose metabolism in particular from a diabetes standpoint. And what we saw was that in these individuals who have relatively high fasting blood glucose but are not diabetic, there was a significant decrease in blood glucose levels, again, over the course of just 1 month. And interestingly, we did not observe changes in fasting insulin. So normally in dietary interventions where you reduce calorie consumption, you also observe a decrease in fasting insulin, and that's not what we saw here. Instead, we saw changes in a hormone called FGF21, which I'll talk a little bit about more later. And basically, it's an insulin sensitizing hormone that some recent research has shown to be associated with protein restriction. And so levels of FGF21 actually doubled during the course of the study. So we wanted to really be able to do a mouse study so that we could look at exactly what was happening. So we could feed the mice exactly specific diets and do much more invasive and prolonged procedures than are possible in humans. And so we first started by using natural source diets, and we placed mice on one of two diets, either a diet with 21% of calories coming from protein or 7%. So 21% is pretty much what the USDA recommended desired protein intake is, while 7% reflects what the type of diet that the humans in the study that I just talked about were receiving. And so these diets were isocaloric in terms of their energy density, and they were matched for fat content. So the difference in caloric consumption is that the 7% protein-restricted mice had more of their energy coming from carbohydrates. And what we did was we simply measured weight and body composition using some facilities that the Department of Medicine has in the VA animal facility, and looked at glucose homeostasis during the course of an 11-week experiment. And so what we found was very interesting. So first of all, we saw a significant increase in food consumption on mice placed on the low-protein diet. And this is in line with both the human studies and some other previous data suggesting that definitely mice and possibly humans can actually sense the amount of nitrogen that they're consuming, so they eat more in an effort to consume more nitrogen. So we looked at weight after 3 and 8 weeks on the diet, and these are young, growing mice. We start at 9 weeks of age, so these are adult mice, but they're still in the process of growing. And so it's expected that they will gain weight. And indeed, they gain about 6 grams during the course of the study. And you can see that the protein-restricted mice gained much less weight, so less than half the amount of weight, even though they're eating more calories every day. We also analyzed body composition. And so what was really cool was that if we looked at fat mass here on the left, you see that the mice gained almost no adipose mass on a low-protein diet. And while we were concerned about lean mass at first, we saw that the mice continued to gain lean mass, albeit at a reduced rate. So these mice eat more, they have less adipose tissue gain during the course of the experiment, and we see that they continue to gain some lean mass. So they're eating more, where is the energy going? Well, as it turns out, the energy is actually going to heat, and so we can assess energy expenditure using a metabolic chamber facility. And what we saw is that during the nighttime, there was a significant increase in energy expenditure in mice on a low-protein diet. So they're actually literally burning off the excess calories. We also looked at some of these metabolic parameters, looking at fasting blood glucose and insulin levels. We saw that there was a significant decrease in both fasting blood glucose and a trend towards decreased fasting insulin as well. If we calculate insulin sensitivity using the HOMA2-IR calculation, we see that there seems to be increased insulin sensitivity in these mice. So one of the things that's easier to do in mice than humans is a glucose tolerance test. And so during this type of test, at the 0 time point here, we fast the mice overnight, and at the 0 time point here, we look at fasting blood glucose levels. We then administer 1 gram per kilogram of glucose, and over the following 2 hours, we assess how blood glucose changes with time. And what we see is that there's a significantly decreased area under the curve on mice placed on a protein-restricted diet, indicating that they have improved glucose homeostasis. So glucose homeostasis is very complex, but glucose tolerance tests assess, in part, hepatic gluconeogenesis. And to remind you, this is primarily carried on by the liver, and it converts pyruvate into glucose that's released into the bloodstream. And so to more directly assess hepatic gluconeogenesis, we can perform a pyruvate tolerance test, in which instead of glucose, we inject pyruvate into fasted mice. And we then track their blood glucose levels over time as the liver produces glucose and it's released into the bloodstream. And what you'll see is that, again, there's a significant decrease in the area under the curve in mice on the 7% low-protein restricted diet versus mice on the control diet, indicating better control of hepatic gluconeogenesis. So we wanted to know what was altered in a low-protein diet that contributes to these beneficial metabolic effects. And so there's 20 different amino acids in the diet, essentially, and 9 of which are essential. So these 9 amino acids are essential in both mice and humans. And a lot of suspicion recently has focused on the possible role of branched-chain amino acids. Leucine, isoleucine, and valine are the branched-chain amino acids, or BCAAs, as I'll refer to them. And these are essential amino acids that have been found to be elevated in the blood of people with type 2 diabetes. And indeed, not only are they elevated in obese and insulin-resistant or diabetic humans, they're also elevated in rat and mouse models of obesity and diet-induced obesity. So this is pretty interesting. They also correlate in humans directly with HbA1c levels, and they can be used as a predictor several years in advance of who's going to develop type 2 diabetes. So we really were interested in looking at these branched-chain amino acids. And another reason that we're interested in focusing on them is we performed an amino acid profile analysis of the blood of the humans in the protein-restricted study. And what we found was that branched-chain amino acids, leucine, isoleucine, and valine, were decreased in the blood of people on a low-protein diet, between 10 and 20% reduction in circulating levels. And what was really interesting was that this was not true for all amino acids. So, for instance, methionine, which is another very metabolically important amino acid, there is no change in circulating levels. And so this suggested to us that branched-chain amino acids were really a good place to look at. There's one other reason to look at branched-chain amino acids, and that's that a lot of people take branched-chain amino acid supplements, primarily for muscle building. There's a belief that there's some studies to support that taking increased amounts of branched-chain amino acids can help you build muscle along with exercise. However, there's also another reason to take branched-chain amino acids, and that's that some people think that increasing the amount of branched-chain amino acids in your diet may actually help improve diabetes. And there's a few studies that suggest that very short-term increases in the amount of leucine in particular can in fact improve insulin sensitivity, but these are all very short-term studies. And by short-term, I don't even mean a month, I mean on the order of days. And longer-term studies have been really complicated and haven't really shown that this is true. In fact, the opposite seems to have been shown. And interestingly, no one has ever explored the impact of reducing branched-chain amino acids on metabolism. And this may be very surprising to you, but it's much easier to take a pill or add something to a diet than to subtract things from a diet. And when people did, back when these amino acids were first being discovered and the first nutrition books were being drawn up about amino acids, when they looked at these, what they actually did was remove amino acids from the diet entirely. And so we have a lot of data that's historical on what happens if you take an amino acid that's essential out of the diet completely. The mice become very unhappy for about a week, and several weeks later they die because it's an essential amino acid and they need to be consuming it. So we have a lot of data on that, but what we don't know is what happens when we just reduce the level of branched-chain amino acids in the diet. So in order to explore this, we constructed a series of synthetic chemically defined diets in which we added a specific amount of each amino acid into the diet with help from Hormonal laboratories based here in Madison, Wisconsin. And we focused on 3 diets at first. We made a control diet and a low amino acid diet, reflecting the amino acid composition of the control and low protein diets that I showed you previously, so 21 and 7% protein. We also made an extra low version with 5% of calories derived from amino acids. And the reason for this was that a number of studies that have looked at protein restriction actually look at the effects of 5% protein restriction. And as I'm going to show you in a few minutes, that may or may not be a good idea. So we wanted to validate that these diets performed similarly to normal natural source diets. And so we looked again at food consumption and weight over time. So both the low amino acid diet mouse groups ate significantly more, just as we saw with both humans and mice on natural source diets. Weight continued to increase for mice on the control diet, but the effects of these amino acid-defined diets were a little bit more severe. And so the 7% diet simply blocked weight gain during the course of the experiment, while the 5% diet actually caused a significant amount of weight loss. We looked at glucose tolerance again, and we see that both restricted diets, 7% and 5%, were capable of decreasing fasting blood glucose levels. And if we look at the area under the curve during a glucose tolerance test, again, we see that there is decreased area under the curve, just as with natural source diets. Importantly, the 7 and 5% diets are basically overlap in terms of both their physical curves and the quantification of the area under the curves, suggesting that 7% is just as good as a 5% diet from the perspective of metabolism. We also performed, again, a pyruvate tolerance test, and just as with the natural source diets, we see decreased area under the curve in mice placed on the amino acid restricted diets. And so due to the fact that the 5% diet mice seem to lose a lot of weight and generally weren't the happiest anyway, we selected the 7% version of this diet, which reflects all of the other experiments that we've been doing, as the basis for our future study. Okay, so at this point, we are finally ready to move ahead and find out what happens to branched-chain amino acids. And so what I'm going to show you next is a comparison between mice placed on 4 different diets, either the control diet with 21% of calories coming from amino acids, the low amino acid diet with 7% of calories coming from amino acids, a low branched-chain amino acid diet in which leucine, isoleucine, valine, specifically those 3 branched-chain amino acids, are reduced by 2/3, such that the level of branched-chain amino acids in this diet reflects that in the low amino acid diet, but the levels of all the other amino acids was left unchanged. And finally, we also looked at a low leucine diet because a lot of data suggests that leucine is actually one of the more potent amino acids in terms of its effects on signaling through important metabolic pathways. And so in this diet, we reduced its level by 2/3 to match the level of the low amino acid diet, and again left the levels of the other amino acids unchanged. So we started learning interesting things right away. So just as we expected, mice placed on the low amino acid diet ate more than mice on the control diet, but so did mice on a low branched-chain amino acid diet. And so this actually suggests that branched-chain amino acids drive a lot of this increase in calorie consumption, or in food consumption during this study. Low leucine had essentially no effect on food intake. When we looked at weight change, we once again saw that weight change over the course of 10 weeks was simply blocked by restriction of all amino acids. And it was also blunted by restriction of branched-chain amino acids specifically. Leucine diet mice, on the other hand, actually seemed to trend upwards very slightly, but was not statistically different from mice placed on the control diet. We analyzed body composition, and we found that just as we had expected there, we blocked a lot of fat increase in mice placed on the low amino acid diet. But if we look at the branched-chain amino acid diet, we see a blockage of increased fat mass. And mice on a low branched-chain amino acid diet also seemed to have decreased lean mass gain as well, although it seemed to be a little bit higher numerically than mice on a low amino acid diet. So to summarize, blocking branched-chain amino acids decreases weight gain during the course of this growing mouse phase and decreases both fat mass gain and lean mass gain. Now, one of the things that we noticed physically while inspecting mice on a low leucine diet was that they seemed very fat. And so this did not fully reflect itself in the body composition data because there seemed to be quite a variance between the mice in terms of how they sort of felt. But we knew that something interesting was going on. And so in this work, we worked with Carolyn Alexander at the Cancer Center to try and understand what was happening to the fat. And so this is looking at dermal fat from either the belly or the back of the mice. And you could tell simply with the naked eye that this intradermal fat layer here, this white layer, is much larger in mice placed on a low leucine diet. We saw a trend towards increased adiposity on the back skin as well. And so when we quantified this, we found that there was more than a 50% increase in the thickness of the intradermal fat layer in mice placed on a low leucine diet. And when we looked at the epididymal white adipose tissue fat pad that mice have, again, we saw a similar increase in the weight of that fat pad. And so the low leucine diet seems to have a significant effect on fat tissue. We don't fully understand why. We haven't been able to assess yet whether this is due to increased adipogenesis or expansion of white cells, but this is something that seems really interesting to us. It also possibly explains some very old data suggesting that low protein diets might actually cause increased adiposity. And we think that that's not true because that's not what we see in the mice. But if you were to eat a diet that was specifically decreased for leucine, maybe you would see some of these changes in adiposity. And so we think that that might be an explanation for some historical data about protein-restricted diets. So we placed all of these mice on an overnight fast and performed a glucose tolerance test on them. And we found that all three diets, the low leucine, low branched amino acid, and low total amino acid diet, all of these improved glucose tolerance very significantly. We once again analyzed pyruvate tolerance. Here we did not see an effect of the low leucine diet on pyruvate tolerance that was statistically significant, but we did see a statistically significant decrease in area under the curve for mice placed on a low branched amino acid diet, as well as the low total amino acid diet. And so this suggests that branched amino acids contribute very heavily to this phenotype due to decreased levels of hepatic gluconeogenesis. But something to draw your attention to here is that there's a very significant difference between the low branched amino acid diet mice and the low total amino acid diet mice. And so what we think is that there's probably contributions from other essential amino acids to this effect on gluconeogenesis and hepatic insulin sensitivity. And that's an area that we're continuing to investigate. I think it's very, you can tell over here, it's very dramatic that the low amino acid diet curve is lower than all of the others. So we also wanted to assess the effects of these diets on insulin and pancreatic beta cell function. There's a lot of data that suggests amino acids can really stimulate or hinder the release of insulin, and we wanted to look into that in some more depth. We saw that there was a trend or a statistically significant result in decreased fasting blood glucose in both low branched amino acid and low amino acid diet mice. But we actually had a very paradoxical result. In the low branched amino acid diet mice, we saw that there was a little bit of an increase in fasting insulin levels. And this was something that seemed very interesting to us, and we wanted to try and understand more about this. And so with Michelle Kimpel's lab, we performed ex vivo analysis of single pancreatic islets to look at glucose-stimulated insulin secretion. So here we either have a very low level of glucose, about 2 millimolar, or a high level of glucose, about 16 millimolar. And we see a significant increase in secreted insulin when you raise the external level of glucose, just as what happens in the body. We actually saw a real decrease in glucose-stimulated insulin secretion on a per-islet basis in mice on both the low branched amino acid diet mice or the low total amino acid diet mice. And this also seemed to be associated with a decrease in islet insulin content in both cases. If you measure insulin secretion, which is simply the secreted insulin divided by islet insulin content, so the percentage of insulin that each islet secretes, there is no significant change. And so this suggests that something really interesting is going on at the level of insulin release. And to try and understand more about this, we teamed up with Matthew Marin's laboratory to investigate the effect of these diets on the metabolism of single pancreatic islets. And so you will have to hunt him down if you want a very detailed explanation of this. But basically, single islets can be perfused with various dyes or transfected with various sensors, which enables the fluorescence detection of changes in various components. And here we're looking at calcium and ATP. And so islets don't secrete insulin continuously. They actually fluctuate, and there's periodic release of insulin. And this is driven by periodic changes in the fluctuation of calcium and ATP. And so what I want to draw your attention to is two things. We're going to look at peak height. So you can see just by the naked eye that we see a significant increase in peak height in low branched amino acid diet fed mice, and an even greater change in low amino acid diet fed mice. And the width of the peak also reflects function of the cells. And so here over on the left, we're looking at plateau fraction, basically the amount of time that an islet spends in a stimulated space that's based off of the width of the peak. And there's a significant decrease in that time, which indicates that these islets have a low workload. So one concern about in diabetes, one of the things that drives the pathogenesis of diabetes, is that islets have to work too hard. And that's not what we see here. We see that the islets actually have a very relaxed workload. And looking at peak amplitude, we see that there's a significant increase in the amplitude of the peaks. As I mentioned, you can see that by naked eye. And this suggests that the islets are being highly efficient. And so probably the reason that they have a low workload is that they're producing insulin and releasing it very efficiently. So rather than there being any insulin and islet defects in these mice, they actually have happier and healthier islets. So finally, I want to talk about the role of the insulin sensitizing hormone FGF21. So FGF21 has been known for a few years now, about 5 to 10, that as a hormone that's induced by fasting. And it's induced by very short-term periods of fasting, like overnight in a human, but more longer-term periods of fasting, such as 48 hours, can result in an increase of this protein by 20-fold. It's also recently shown, just last year, to be induced by a low-protein diet. And this particular study used the really low 5% amino acid diet that we showed caused severe weight loss in the mice. So this is actually one of the reasons we avoided this type of severe intervention. And we want to know what happens to FGF21 in a more moderately protein-restricted diet. And so one of the things we looked at was here on the left, we looked at circulating levels of FGF21. And we see that there's about a 3-fold increase in the circulating levels of FGF21. You'll note that there's really big error bars. That doesn't reflect anything other than biological variance. So the levels of circulating FGF21 have a very large amount of variance, even in genetically identical animals fed the same amount of diet and looking at them after fasting for the same amount of time. We also looked at FGF21 gene expression, which correlates very well with levels of circulating FGF21. It's believed that FGF21 is normally produced primarily by the liver, but it can also be secreted by other tissues, including muscle and adipose tissue. And so we looked at levels of FGF21 mRNA. We see, again, that there's a significant increase in FGF21 in the liver, and there's also in the muscle and adipose tissue. Whereas, interestingly, in the low branched amino acid and low amino acid diet mice, there's no change in circulating FGF21 and no significant changes in the mRNA of FGF21. FGF21 is actually responsible, or believed to be responsible, for the increased heat expenditure of mice placed on a low-protein diet. And so we looked at heat expenditure in these mice on our special diets. We see that there was a significant increase in the low amino acid diet mice, consistent with the increased levels of FGF21, but no statistically significant change in mice on the other restricted diets. And then finally, it was recently shown that FGF21 stimulates the release of another insulin-sensitizing hormone, adiponectin. And we see release of adiponectin in low amino acid diet mice, but no increase in adiponectin in low branched amino acid or low leucine diet mice. So all of this data together shows us that there really is no change either in serum levels or downstream indicators of FGF21. So the results we're seeing are through a novel and probably very interesting mechanism. So we're still working on figuring out what that mechanism is. We're taking a phosphoproteomics and metabolomics-based approach using some new facilities and resources that Josh Kuhn's lab has opened at the UW Biotechnology Center. But we have looked at some gene expression data, and one of the things that we found that was very interesting is that all three of these diets, low amino acid, low branched amino acid, and low leucine, seem to coordinately regulate pyruvate metabolism in the liver. And so this is very consistent with the fact that we see altered glucose and pyruvate tolerance and altered hepatic gluconeogenesis, as all of these genes that regulate what happens to pyruvate are being altered. And so we're working on trying to figure out what the metabolic and physiological basis of these effects are. Okay, so finally to wrap up, to conclude, we have very good evidence of decreased protein in humans and mice and decreased branched amino acids in mice improve metabolic health. We also have really convincing data suggesting that whatever the mechanism is by which low branched amino acids improve metabolism in mice, it's not through alterations in circulating FGF21. And finally, we are just beginning to scratch the surface of how dietary amino acids and other factors might explain the beneficial metabolic effects of a protein-restricted diet. So branched amino acids definitely explain some of the effect, but we're really interested in finding out what all of the essential and non-essential amino acids do in regulating metabolic health in mice and eventually in humans. And so that's our first future goal. We're trying to launch a study drilling down at the other essential amino acids and looking at what happens when we decrease the level of those essential amino acids to metabolism and physiology. We expect to see some really interesting results based on our surveys of the literature thus far. And we really want to understand the molecular mechanisms that mediate the effects of protein restriction overall, as well as specifically altering branched chain amino acids, on the theory that this may give us an insight into novel metabolic pathways that could be developed into drug targets. We'd really also like to know if reducing branched chain amino acids might be a sustainable and translatable therapeutic intervention. And so what we're hoping to do is decrease the levels of branched chain amino acids in mice that have been preconditioned to be obese and diabetic and test that. And in the longer term, we hope to be able to use medical foods that have been developed for maple syrup urine disease which are basically free of branched chain amino acids to be able to test whether reducing branched chain amino acids could actually be an intervention in humans. And so hopefully sometime in the next year or so we'll be able to do that. And finally, my own background, I didn't exactly come into this as an endocrinologist, although one of the great things about working as an endocrinology division is that there's a very heavy endocrine focus and a lot of resources and help in looking at endocrine things. I'm really started as an aging biologist, and so I'd like to know if reducing branched chain amino acids could, much like a protein-restricted diet, increase both health span and longevity in mice. And so to summarize, we may want to rethink taking branched chain amino acid supplements or possibly any dietary supplements, because we're really just at the beginning stages of trying to figure out what these do. But it seems from our data fairly convincing that taking branched chain amino acids from the perspective of diabetes can only be bad for you. And so we might want to concentrate instead on some other foods. So we are omnivores, and so certainly you could advise yourself to have bananas, orange, and pecans. Those all sound healthy, and those are all low in protein. But there's a lot of unhealthy sounding foods that also have low protein. So Nutella, for instance, is about 6% protein. Ice cream is about 4% protein. Interestingly, just in terms of the foods that's available at Grand Rounds, bagels have the most protein. And so even though they sort of seem like a healthier choice than a cream donut, from the protein perspective, a cream donut is actually a much better choice than bagels. And if we took this to its logical conclusion, of course, we might really focus our diet on breakfast cereals, where if you don't have milk, less than 4% of calories come from protein. And so I'd like to thank everyone who was involved in doing this work, particularly my laboratory, Nicole Cummings, a graduate student in the Endocrinology and Reproductive Physiology program, did most of this work, or spearheaded most of this work. Everyone in the lab has contributed. And our great collaborators, Calorie Alexander and her postdoc Ildiko, Luigi Fontana at Washington University in St. Louis and his clinical trial team, and Michelle Kimple at Matthew Marians and their laboratory members who've contributed to the islet studies. And also our funding sources, the NIH, the VA hospital for providing facilities and resources, and several other groups who have supported us, including, of course, the Department of Medicine and the Division of Endocrinology, which enabled us to have the equipment to look at body composition and energy expenditure. Thank you.
Thank you. I'll ask you to call on the audience, and we have a microphone, so whoever wants to ask a question, please raise your hand, and we'll try to pass that to you.
So, Dudley, I was interested in the experiments and their effect on the skeleton. We've known for a while that high-protein diet increases urine calcium, and that was thought to be an indicator that it was causing harm to the skeleton. But it turns out that high protein increases intestinal calcium absorption and actually makes greater calcium available for skeletal mineralization. So when you're giving these restricted.
So the question is, what happens to the skeleton? And we don't know. We've only started our physiology investigations. So one thing that we do want to do is we want to focus on muscle and musculoskeletal performance and trying to understand what the effects of reducing the amino acids are in that context. But we don't really have very much expertise in looking at the skeleton. So if you want to help us look at the skeleton, that would be terrific.
Thanks. Dudley, that was a terrific talk and very engaging. So I have a question, which is one of the most challenging things about what you said for me as a primary care doctor is this concept you discovered where these rats and humans will increase their caloric intake and yet lose weight. And what we tell patients in the office is it's calories in versus calories out, and Atkins only works because you take in fewer calories. And do you have any sense as to how in the world it could be that these animals and humans could be consuming more calories and yet losing weight?
And Dudley, may I ask you to use this microphone? Just we're having a bit of technical issues. Thanks.
So that's a great point. Calories in, calories out has definitely been dogma for a long time, and I think that is something that our investigations and other investigations at the NIH and elsewhere have shown that may not be quite true. So there's a recent experiment where they actually put humans in metabolic chambers at the NIH, sort of a difficult experiment. You have basically several rooms rigged as metabolic chambers. And what they showed was feeding the mice different diets. I think they concentrated on carbohydrates. They were able to show that the dietary composition changed energy expenditure and weight gain in that context as well. And so what we think is that the extra calories are primarily being burned off in the form of energy. We think that white adipose tissue is probably beiging, or brown adipose tissue is being activated in the protein-restricted context. What we don't really understand is what's happening in the branched amino acid restricted context, because there we don't see the energy being burned off, but we do see the increased calorie consumption. And so one possibility is that we need more statistical power to sort of sort out those differences, or something else interesting might be happening. So the microbiome, for instance, seems to be very involved in regulating how well we metabolize food and how we take it up. And so we've just started working with Federico Rey's lab to look at the microbiome of these mice. And so hopefully we'll be able to find out if there's something there that's happening as well.
Has anyone looked at the impact of protein restriction on IGF-1 and IGF-2 levels, and do you think it might be useful to look at that?
So protein restriction decreases levels of circulating IGF-1 in mice, but not in people, or at least not in the study that we looked at. It does, however, in humans seem to alter levels of IGF binding proteins, and so the bioactivity of the IGF that is circulating may be different. And we think that the IGF changes in circulating IGF may drive some of the other effects. So based on some mouse studies that we did with Luigi Fontana's laboratory, we showed that protein restriction slows tumor xenograft growth. And we think that decreased circulating IGF may be one of the things that drives that. But I think it would be really interesting to sort of pursue IGF and other related hormones to see if they are involved.
Hold on the microphone, please.
The effect of increasing leucine as a single amino acid?
So we haven't looked at the effects of increasing leucine yet. One of the drawbacks to this type of essentially nutritional geometry-based experiments that we're doing is that there's a lot of different groups and combinations to test. So one of the things that is on our radar is the idea of what happens if we were to, say, double or triple the amount of branched amino acids. And if we saw effects there, then probably we'd go and pursue single amino acids like leucine as well. One of the other things that we don't know yet is what the effects of decreasing single amino acids in the context of isoleucine and valine are, and that's something that also could be very interesting.
Okay, well, I'm wondering, with respect to the lifespan and health span comment you made at the very end, the caloric restriction paradigms that have been out there for a long time and the potential benefits on various disease processes, in those types of scenarios, what types of adjustments of the three elements were made to bring down the calories that produce those benefits?
Sure. So typically speaking, a calorie-restricted diet where you reduce caloric intake by 40% is simply done by reducing the level of each dietary component by 40%, and then usually spiking in some extra vitamins and minerals to make sure that there's enough micronutrients involved. So although technically speaking it is not always the case that calorie restriction means protein restriction, almost all calorie restriction interventions are also protein restriction interventions. And so this was something that was investigated a number of years ago as something that might be very interesting, and people got conflicting results probably because they were trying very low protein diets. And so some of the rat studies that were done involved 4% protein diets that eventually seemed to really harm the animal. And so I think that was sort of ignored for a while, and now we're trying to look at more moderate interventions that better reflect what would happen, say, in a calorie-restricted individual.
Hi. I wonder, when you're trying to figure out the biomolecular mechanism involved in it, I know that adiponectin and IGF can tie into the enzyme AMP kinase that is activated in low energy states. And the one picture that you showed that you had a dramatic decrease in the ATP and an increase in the calcium could actually go along with the activating mechanisms of that enzyme, which include, again, low energy, low ATP, high AMP, and also which makes it more available for activation by LKB1, but also calcium calmodulin can activate it as well. So I just wondered if you've looked at all at that.
So yeah, so the idea is that, in terms of what pathways are affected. So we've looked at a number of pathways in a number of different tissues now. And so what we do see is that we do not see activation of AMPK or increased phosphorylation of AMPK substrates, probably due to the fact that this type of intervention is not severe enough to trigger those changes. And so fasting or prolonged fasting seems to be, and we think that if you lower the protein content enough, you would see AMPK activation, but we don't see that yet. The two major amino acid sensing kinases in both mice and humans are mTOR, which is also downstream of AMPK, as well as downstream of amino acids and IGF, and GCN2, which is both downstream of mTOR and is regulated by uncharged tRNAs. And so those are sort of where we're concentrating from the kinase aspect, but there are also a lot of other biological processes. They're probably affected by amino acids and enzymes or activities that bind to different amino acids. So we're concentrating right now on the kinases, and we have some interesting preliminary data, but this is going to be sort of a long-term project to fully work everything out. Yes.
Thank you. Okay. Oh, great. So NIH is very interested in looking at sex differences now in preclinical studies. And of course, one could think of a number of reasons why your male and female mice might have differences in the estrous cycle or whatever. So anyway, I'm just wondering, did you look at sex differences in the effect of your intervention?
That's an excellent point. We're really interested in looking at sex differences, and so a lot of our other projects we're looking at both male and female mice. Right now, we've only started with males, so the inbred strain of mouse that we're using, C57 Black 6, typically you can get away with using fewer animals because in terms of metabolic and particularly glucose responses, there's a larger dynamic range. And so that's where we've concentrated. But in terms of the proposals that we're putting into the NIH and others to sort of continue this work, we hope to be able to look at both males and females and also to test our interventions in both males and females. Yes?
Dudley, I think you mentioned that the low-protein diet caused the mice to have greater activity at night. Is that true? Do you have any theories about that, or are they just not looking for food?
So they do not have increased activity at night. So our metabolic chambers allow us to track both respiration, wheel running, and motion within the cage. And so normally mice are nocturnal, and so they move a lot more at night than they do during the day. And so typically speaking, they eat between 60 and 80% of their food at night. What we were looking at was energy expenditure, which is actually it performed by indirect calorimetry, basically comparing the amount of oxygen that comes into the cage and oxygen out, and CO2 in and CO2 out. And so we saw increased energy expenditure at night. They are moving more, though, but there's no difference between the control groups and the dietary intervention groups in terms of physical activity.
We have time for one more question and one more answer. That's it.
So another primary care physician, kind of practical question. What is your recommendation for us as primary care docs for our patients? I've started to switch to more of the Mediterranean diet, and then protein shakes are extremely popular, and really this shifts my thinking about all of that. Do you want to comment on what would be good advice for us as primary care physicians?
I'm going to stick with Michael Polland and say that you can't go wrong with not too much, mostly plants. But I think that protein shakes in particular, unless someone is, really actively involved in a career as a bodybuilder, I think that probably that all of the works to date suggest that those are to be avoided, and those probably would increase disease risk, both for cardiovascular disease, cancer, and diabetes.
Great. With that, I've got to say it's got me rethinking our food out at Grand Rounds. I used to recommend bagels, and now I'm recommending donuts. I don't know. I want to thank Dr. Lamming for just an outstanding Grand Rounds.